Okay, I hope you can hear me, and thanks very much. I wish I could be there. I used to work across the street from the building you're currently in at the EBI as a group leader in research, and also a special shout out to Tim Hubbard, whom I met at the time, and others whom I know in the audience. And I want to tell you about some recent work that we've done that relates to the risk of cancer. And this is an effort that tries to catch cancer early. And the way we do that by looking at clinical records from patients to then predict the risk of getting cancer before they get cancer. And for this, of course, you have to train. If you're successful, you can actually design surveillance programs where people are looked at in great detail, and then early intervention to actually do something about the cancer before it gets too bad or even before it arises. This was very much a collaboration, coming together different institutions, both from Copenhagen and Denmark. That was very important. And then the Cancer Research Institute, Dana-Farber, Veterans Administration, MIT, Harvard, and funded by philanthropy, and also another contribution from Denmark, from the Novo Nordisk Foundation. The challenge is that pancreatic cancer, such as well as ovarian cancer and others, are very aggressive. They're detected, typically 20% are detected in early stages. You see that on the left. And those actually have a much better survival, 5-year survival. But most of them actually have a survival that actually is abysmal. And therefore, the point is, if one can actually detect the risk of cancer early and do surveillance programs with new technologies to detect the cancer early, like from blood samples, like some of the recent work from the UK Biobank that you may know about. And then, of course, one can actually resect the cancer and move more patients into an area where they have better survival. So catching cancer early is the goal, and that very much has to have clinical implications. To actually successfully reach the clinic, you have to not only have risk prediction, which is what we worked on here in the first step, but then you have sensitive detection methods, for example, from bodily fluid, which would be ideal, or some imaging methods, which can be expensive. And then you have to intervene by either doing early therapy, could be surgery, other kind of therapy, or even better yet, prevention of cancer for those people who are at high risk. That's the clinical application. We tested that by accessing very large electronic health records, as is essential to be trained, partly to cut through the noise in clinical records. In Denmark, 6 million US Veterans Administration, which is one of the best organized in the US, which otherwise is very fragmented, 10 million patients. So we train on a lot of patients, a small subset of which have the particular cancer, pancreatic cancer, and then you have to test this on withheld data. The databases are essential, and it's very interesting and complex and challenging that the distribution of in terms of trajectory length and also time of availability of clinical records actually is different in different healthcare systems, so that becomes an issue. So the data quality matters a lot, and you have to have an access to a confidential behind firewalls, a GPU cluster, and that's a challenge that has to be solved. There's one aspect of getting to clinical applications. The machine learning you're all familiar with, let's show briefly. The innovation that we used, other than beyond well-known AI methods, is to use time trajectories of patients' disease, one of those 6 or 10 million, the actual history going back some years, not just whether or not they had a certain kind of disease. And then when you make the prediction at a certain time point before they have cancer, to actually predict whether or not they're going to get cancer within 6 months, 12 months, or 3 years. And so there are 2 time aspects to that, and that affects, of course, how you do the machine learning. The analogy is natural language, where you have a system of words, and then you try to see what comes next. So similarly, disease events, this is a time axis, the black bars are disease events, the question what comes next. And we formulate that because of the time element that we want to capture as a probability curve over up to 5 years or 3 years, what's the likelihood to actually get cancer? No cancer, 0, 0. And then in the third year, somebody got cancer. So we train on data that is binarized in this fashion, but over a time sequence. And that is quite beneficial in terms of designing an intervention program. It takes into account whether or not somebody is at risk of getting cancer very quickly or over a longer time plan. The training is done on these large databases, and as you're familiar with, you have to have a training set, maybe a development set to develop optimized hyperparameters, and a test set that's withheld, you never look at least in theory, you never look at it. It's all withheld, and you do test on that. Retrospectively, you test on that withheld data set. Ideally, you would test those methods prospectively, but this is the best way, a withheld data set, to test whether or not something actually is working on the data system in which you've trained it. And so what we're using is clinical histories. These green circles are disease events, cancer occurrence or not. And so these time elements are taken into account. You put that through a transformer, and then you optimize a loss function, which here is the difference between the observation, which is binary, and the prediction, which is a slowly rising probability curve, so that this gray area is minimized. So that's how you train. It's supervised training. And the key element after transforming things from a discrete state into real number vectors is this encoder. And even though cancer diagnosis is a binary event, what we learn actually and what we train on is the actual time sequence in terms of the probability curve. So supervised training on those data sets is tricky because out of a large number of people, you only have maybe 0.4% who actually get over their lifetime but get cancer. Once you have a method, get that particular cancer, pancreatic cancer. Once you have a method trained, then you can go to prediction, and you can apply this now to a very large number of people quite inexpensively. You use the model to predict whether or not somebody is getting cancer, and you have to pick an operational threshold. People above that are at high risk, below that they are not, even though you can also work in terms of a gradation of risk. And then the challenge is to then proceed with those at high risk and do clinical screening. We pick a fairly small number as high risk. Why? Because for something like pancreatic cancer, the detection methods are not very well developed, and so only expensive tests like MRIs, endoscopic ultrasound, and so on are available. So you want to start with a small number to test out these methods until eventually you can get to population-wide screening when prediction gets better and when the testing methods become less expensive. So contrary to mammograms, initially we focus on which is possible in a large population, we focus on those at highest risk. So technically we ask some questions that what methods works best. Do we have a method that will transfer from Denmark to the US, for example? And what would the scope of a surveillance program be, as I just mentioned? So technically, transformer is the best one of several methods. It's harder to make a longer-term prediction whether somebody gets cancer within 3 years. It's hard to predict whether somebody gets cancer within 12 months. What do you use as input? Disease codes. We now have new work that I'm not showing right here. It's about to be submitted. Adding medications, actually, in addition to disease codes, which are standardized internationally as our medication. So more and more data is better than less data, obviously. And then, of course, it is harder to explain, to predict whether or not cancer occurs in a larger time interval. So those are some of the technical questions. And we focus then, typical report is area under the curve, what it might be. And you can see, 1.0 is perfect, and this is random. So you want to be up in the upper left-hand corner. You know these curves, true positive against false positive rates. And that's interesting if you want to compare different methods. But if you want to apply something in real life and make a prediction, you have to pick a decision parameter somewhere along those curves. So we focus on the left area here, which is low recall. Why? Because we want to reduce false positive rates. That in the initial stages, because as you roll this out into the clinic and convincing clinicians and working with clinicians to do this, you want to make sure that actually the false positive rate is low and the positive predictive value, PPV, is as high as possible. So there's an art of how to get that right in the usual confusion matrix that you're familiar with, and you optimize the decision point that you use in real life. The best way to report this, I think, is actually to think of a large number of patients, say 1 million patients, and then along this curve, you take smaller and smaller and say take 1,000 out of 1 million, and then you can ask how good is the prediction relative to what you would do if you just take into account the incidence in the population without extra information, maybe stratified by gender and age. So we are about a factor of 30 or 70 better, depending on the healthcare system, and this also reflects the quality of the data. And there's ongoing work. We've now proved it by adding medications, and one also would like to add blood values and so on. There's technical work to be done in the future, and this, of course, calls for collaboration. One can assess whether or not somebody is at high risk, whether or not they're likely to actually get the cancer, within a short time or a longer time. And you can see that if you look from in the 3-year period, actually there's a decrease in the information content, and this is important to analyze by about a factor of 4. So it's very important in these prediction methods and application, and to develop trust. It also has to do with interpretation, to make sure to assess where is the information content in this case in the clinical records, and where is the information that's in terms of actually being able to predict the time interval at which the cancer occurs. Then of course coming to interpretation of another kind, to look at features. As you know, this is complicated. You use something like IG gradient or something called the Shapley method. It is not easy because out of 2,000 disease codes, we use only a small number actually have a high predictive value, and some of them are quasi-symptoms like unspecified jaundice, or of course if somebody has pain and so on. The art is you're training on 2,000 disease codes, and then you report using one of these methods the isolated contribution of individual factors, and that's technically actually quite challenging because machine learning does things in context, while this is picking one out at a time, and each one alone does not in an additive way together with others then predict the occurrence of cancer. So that's challenging, and that's of course ongoing technical work in the field. We have some data also in terms of predictive value for black and white, and male and female. It turns out for white persons in the U.S. Veterans Administration system, the predictive value is higher. For females, slightly higher than for males. You would learn the bias, look up the bias that the machine learning method has derived from data sets which are uneven, mostly because of socioeconomic differences. And so to then do that better in the future, once you know where the differences are, where the potential bias is, you have to actually do several things. Either you change, either you train separately on different subgroups, say black and white, if it's ethnic or socioeconomic, and or to change the machine learning method not to learn those distinguishing features that are not predictive of cancer, but actually take those out and get a method that will predict with even accuracy across different groups and reflect the difference of occurrence of cancer in those different groups. So that's a technical challenge, so that we are currently working on, and are others. You can look this up in a publication in this journal here. Also, there was a news clip on CBS News that actually became generated some public interest in the US this one year, PDAC risk-CBS. And importantly, for us, and I think maybe for this kind of effort, we make this software available in an open source fashion so we can generate collaborations. Lots of challenges in this, which I can't go through right here. But most importantly, it's important to remove the bias that's inherent in these methods because of uneven databases and, of course, uneven healthcare systems, and then to actually access to the right kind of data sets and computing resources, which is quite non-trivial. Done very well in Denmark and the Vegetable Administration. Others are in the works in Germany, for example. In the UK, I talked to Tim Hubbard about that some time ago. UK Biobank, we already have access. Others, that's going to be a major opportunity because the UK has one of the best organized healthcare systems. The other challenge is to accelerate this in a way that gets to the goalpost in a small number of years rather than a longer time. And so I plan to collaborate with others by using a network effect, perhaps using one of the grand challenges from Cancer Research UK, the other organizations. And the important thing is to collaborate with your friends or collaborate with people who are highly open to sharing. And I think that's a major challenge of how to get there. You may find it interesting to look at this book, Early Detection, that just came out by a philanthropist who talks not only about the scientific problem, but also about the economic and social barriers one needs to lower in order to be able to get to early detection of cancer. If it's successful, of course, that will be of major benefit, we hope, to patients, and also it should bring down the cost of cancer care. People will die, but they'll die of something else. But cancer treatment is very expensive and wants to bring down the cost of that. And that's an important aspect. So overall, we want to build an alliance that, having worked on this step one that I just reported on, ongoing work, to work with others who do sensitive detection methods, such as mass spectrometry or antibody-based methods, as tested recently in the UK Biobank cohort. Very exciting new developments to be established, but that's also absolutely essential. Once you have a high-risk population, you want to be able to detect the cancer in those. Some false positives, yes, but with a reasonable subset of those patients, you want to be able to actually detect the cancer as early as possible, and then either have early therapy or other kinds of intervention. So I hope to build an alliance going beyond this particular collaboration, and I'm very interested to talk with others. I talked to the Early Cancer Institute in Cambridge, which is a very interesting organization. You may know about them, and that's it. Thank you very much.
Awesome. Thank you so much, Chris. So we are open for questions, maybe one or two questions from the audience. Priyanka? Yeah. Maybe while she's bringing the mic, I have one question, Chris. I think these are like very interesting in like if you aggregate it, as you said, across multiple centers and then you keep track of patients, and I think that would be kind of like a really good data to feed into like large language models and take the text representation from them. But then this type of data is also notoriously dirty in that sense. There are like a lot of missing values. Not everywhere is like not all the data sets are like UK Biobank, so they're like a lot of missing values. And then, so how do you think about like what do you think about like handling these missing values and then feeding them into the modeling and like, or like the domain shift and domain effects that might be stemmed from these differences?
Yeah, that's a good question. We see huge differences between different healthcare systems with the accuracy of the data. I could go on and tell you detail that's just really quite striking, where even the cancer diagnoses are inaccurate. You have to have a cancer registry to look at that. Missing values, yes, there's quite a bit of that. Time distribution is uneven. A main thing to do is to use the different types of information jointly. Mm-hmm. So, for example, if you have disease codes and somebody gets a diagnosis and maybe that's inaccurate, you may want to look at the actual records after the diagnosis of cancer and look whether or not the treatment they got and the procedures they got are consistent with the diagnosis. We also are starting to do, as you just mentioned, natural language processing. It doesn't have to be very complicated in terms of large language models. Mm-hmm. If it is large language models, they should be specific to the domain. Mm-hmm. But fairly simple extraction of the relevant features from medical notes is one way to do it, and that's work in progress. The important thing is to capture in a clinical setting the type of information that's more reliable because of medical ethics. Clinicians will typically maybe enter the wrong billing code, in the US in particular, different in the NHS, but they will not necessarily prescribe, and they won't do unnecessary surgery. So together, these different data types, you can develop some methodology that extracts the most reliable data items. And in the same way, if you now interpret those features and you want to work with clinicians, convince clinicians that this is something that's worth doing, then the interpretation matters. And even though interpretation is technically difficult of the contributing features, it's important to develop trust among clinicians. And for example, pancreatic cancer, they know that diabetes type 2 and obesity and unexpected weight loss are quite predictive. So you want to convince them that machine learning takes that into account, and we can do that partly with the best possible interpretation methods.
Awesome, thank you so much. And any question from the audience?
Okay. So you mentioned some of the challenges, and the countries that face the data infrastructure challenge are also the ones that get hit by the socioeconomic and racial bias. Do you have any thoughts of how we can work towards overcoming those issues?
Yeah, I mentioned that briefly, and I went to a conference where this was the major topic of a whole conference. This was in the US, and there's a lot of interesting work going on. The difficulty is that the typical studies are epidemiological on relatively small groups of patients. So we're now testing this methodology on millions of patients to look at those differences. How do you address it? As I mentioned, one can put into the machine learning, this is my favorite solution, terms into the loss function that actually says, do not predict the difference between black and white or male and female, but predict only the cancer occurrence. And it's possible to do that, we think. It's a work in progress. Either that or you train separately, and then you do it in a way that actually evens out the bias in the prediction system. And as I said, it's important, though, to make sure that you capture the actual real-world differences. For example, higher occurrence of pancreatic cancer in black versus white and other cancer types. Black, white in the US is important as much as socioeconomic. So you have to capture that realistically, at the same time for the AI not to predict whether or not somebody is white or black. And it's a subtle difference. But I think it's technically possible, and I think that's the best way to do it.
Awesome.
Thank you.
I have one more question based on what you said. So there will be actually like a lot of challenges with the biases, right, for these models. They might be, so I think debiasing would be another issue here. Yeah, I think cool direction. Yeah.
I mean, there's, as I said, there's bias in the healthcare systems, obviously, and it's more striking in the US, I think, than in other countries. But so there are real differences in the healthcare practices and application and prospects and so on, and there are also biases in the prediction methods, as we heard, like the images analysis. So one has to separate the two, remove the bias that's inherent in the AI methods because it trains on uneven data sets, and get to a point where that's removed, but the real differences remain.
Awesome. Thank you so much, Chris.
Okay, thank you.
Yeah, have a nice day.
I wish I could be there another time.
Yes.